Okay, it is just after 5:30, so we will go ahead and get started. Aloha, and again, welcome to a special presentation, Molokini during COVID-19, a look at research conducted at the island during the height of the pandemic. We're all very excited that you are here. I am Jill Wirt, your MC, and I'm a project manager at Maui Nui Marine Resource Council. Tonight's presentation is part of Maui Nui Marine Resource Council's monthly Know Your Ocean speaker series, usually held on the first Wednesday of each month at 5:30 p.m. via Zoom, except for tonight. It's the last Wednesday of September, but thank you all for being here. Maui Nui Marine Resource Council is celebrating 14 years of working for clean ocean water, healthy coral reefs, and abundant native fish. This monthly series is supported by the County of Maui Office of Climate Change, Resiliency, and Sustainability. A few things before we get going. You'll notice that your microphone is on mute, so please keep it on mute during the presentation to avoid any distractions. We invite you to submit any questions by using the Q&A button on the lower edge of your screen, and we'll leave time at the end of the presentation to answer those questions. If you're tuning in from Facebook Live, feel free to leave your question as a comment and it will be relayed here through the chat. So now it's my pleasure to introduce our presenters. Russell Sparks is an aquatic biologist for the Maui District Office of the Division of Aquatic Resources, also known as DAR. He's worked for the Maui DAR since 1998 and has helped develop the Maui Marine Monitoring Program. He's the DAR biologist in charge of managing the Molokini MLCD, or Marine Life Conservation District, and has worked very closely with marine tour operators over the years to implement vessel passenger reporting, and more recently to coordinate schedules to help reduce crowding and other concerns within the MLCD. Kevin Weng studies the spatial ecology, migration, and habitat requirements of fishes and seeks to provide scientific guidance for management and conservation. Present work includes studies of reef fish life history to inform size limit regulations, the effects of human presence on fish communities in the context of tourism, and participation in the Holomua Marine 30 by 30 initiative. He works at the Virginia Institute of Marine Science at the College of William and Mary. Whitney Goodell is a marine ecologist and geospatial analyst with National Geographic Pristine Seas. She is based in Honolulu, Hawaii, and earned her Master of Science in the Fisheries Ecology Research Lab at the University of Hawaii at Manoa in 2015. She uses tools such as underwater surveys and remote cameras to observe biological communities of the ocean, from shallow nearshore habitats to the deep ocean seafloor. By evaluating biological information in a spatial context, Goodell builds a better understanding of patterns and relationships that drive biological distributions in the ocean, and applying this information to improved stewardship of our marine ecosystems. Over the past 40 years, Dr. Alan Friedlander has spent over 12,000 hours underwater, from coral reefs to the poles and to the depths of thousands of meters. Dr. Friedlander is chief scientist for the National Geographic Society's Pristine Seas program and a researcher at the Hawaii Institute of Marine Biology at the University of Hawaii. He leads research efforts to help understand and conserve iconic special places in the ocean and is an expert in marine ecology, fisheries, and conservation. Alan received his PhD from the University of Hawaii and was a National Research Council postdoctoral associate. He is a fellow of the Royal Geographic Society, fellow of the Explorers Club, and along with the National Geographic Pristine Seas team, was awarded the 2014 Environmental Hero Award by the Environmental Media Association and the 2016 Crystal Compass National Award from the Russian Geographical Society. Dr. Laura Gajdzik is a marine biologist and works as a biometrician for DAR in Honolulu. Her scientific background is in fish ecology, specifically about coral reef fish. During her latest postdoctoral research, Laura used underwater surveys, including scuba diving and environmental DNA sampling, to assess biodiversity patterns in coral reefs of the Red Sea, and she worked on how to include climate change information in the design of marine protected areas. At DAR, her role is to provide statistical support for the analysis of marine and estuarine data from the main Hawaiian Islands to better manage our aquatic resources for the generations to come. This Molokini study could not have been possible without each of these critical team members, so thank you all again for being here tonight. I'll go ahead and hand it over to Russell and let the scientists take it from here.
All right. Well, good evening, everyone. Thank you for joining us. The Division of Aquatic Resources has been working for many years to better understand numerous things that affect the marine ecology. We oftentimes work on fishing regulations, but over the years, as more and more tourists have been coming and there's been more and more people out in the ocean, the question about the impact that heavy human use in certain areas might have on the marine ecosystem has become more important. And we've been looking at this and trying to get a better handle on managing human use at Molokini and other really popular places for quite some time. Many of you may have recalled in the past, we did some work, social carrying capacity work at Molokini. That was around 2010. And it got a sense that the people who go there were feeling crowded and they were experiencing conflict with other users. And then in 2015, we did some work with Dr. Friedlander and one of his students, Alex Phyllis, and we did tagging work and found that some of the reef predators were being displaced when there was more vessels and people in the crater. But the one thing we weren't able to do is really turn on and off the amount of people at Molokini. It was more when there wasn't too many people there, we could passively collect some data, but usually that was because the weather was really rough and boats weren't going there. And as a result, we weren't going there either to get in the water and look. So the acoustic data we were using at the time would still collect data, but we weren't able to really get a look and see how the entire ecosystem was responding in relation to human use patterns. Well, lo and behold, if I take you back a few years, on March of 2020, we encountered this unprecedented pandemic, right? And pretty much overnight, people were told to go home, try to figure out how to work from home. Tourism to the state dried up. Anyone wanting to come to the state had to endure a 2-week closed in their apartment or their house quarantine. And actually for a while, we weren't even allowed to get out and do field work. So it was during that time, I think probably late March or so, that I was talking with Whitney for some reason. I'm not sure why we were talking, but I was kind of bemoaning the fact that it was such an ideal opportunity to get out and see what was going on at Molokini. And we weren't able to get in the field. And we weren't able to really pull together a team of experts very easily either. The division was, like most state agencies, were looking at probably mandatory furloughs or layoffs. The budget was really uncertain at that time. We didn't have money to pay for anything. There wasn't a lot of grants available for research. People were just focused on the pandemic. But the discussion continued. Whitney talked to Kevin. Alan was dragged into the discussion. And at some point we figured, hey, these guys are all stranded in Hawaii too. Alan's normally off in Antarctica or someplace else, along with Whitney. Kevin's oftentimes in Virginia, but they were all in Hawaii. And it seemed like everybody really wanted to try to find a way to get over here and do the work. Lo and behold, we reached out to the Maui Nui Resource Council. They put out a call for donors, and they were able to get money donated through a GoFundMe operation. Many of you may have donated to that, so thank you very much if you did. People donated apartments for the researchers to stay in, vehicles for them to drive, airline tickets and miles to get here. And we were also able, luckily, to get a travel voucher. We convinced the governor that this work was in fact critical, and they were given travel vouchers to fly over here and avoid quarantine, at least during the day when conducting the work. They were locked back up into the apartments in the evening and not allowed to go out and do anything. I had to bring them food so they could survive on a few occasions. But anyway, we pulled it together, and luckily, because of all of your support and the Maui Nui Resource Council, we're able to do this work starting, I think, in April, and then coming back again in November, So April of 2020, back in November, around November of 2020, and then again the following year, April or May of 2021. And that, of course, across that time, there was a huge gradient in human use, from no activity initially to moderate activity in November when some people were coming back, but they had to test. And then to pretty much back to normal or more so in the following May of 2021. So without any further ado, I want to go ahead and turn it over to Alan and Kevin to walk through the presentation, and then we'll be sticking around and happy to entertain some questions at the end. So take it away, Alan.
Unmute. Aloha all. Hopefully everybody can see that screen. So give me a thumbs up, Kevin, if you can see it. Great. Okay. So thanks for everybody for coming tonight. And again, Russell alluded to the fact that this couldn't have been done without all the support of Maui Nui Marine Resource Council and everyone else. And this was such an unprecedented opportunity that we want to share the results relative to what we found at Molokini, but also I want to give a little background of marine resource use in Hawaii and the previous work that we've been doing in Molokini over the last couple decades, actually. So it kind of goes without saying that we need healthy oceans in Hawaii, but it also depends on your perspective. So healthy oceans mean different things to different people in Hawaii, whether it's for cultural purposes, for sustenance, both non-consumptive and consumptive commerce, any kind of recreation you can imagine, we do in Hawaii, just the pure aesthetic value, the science. You know, Hawaii is unique. It's got almost more endemic species found nowhere else on Earth than any other marine ecosystem. So it's an extremely unique and special place, as we all know, and it's really valuable to all of us, and that's why we're on this call. Although there's a lot of threats posing the reefs in Hawaii, and those threats aren't unique to Hawaii. They're pretty universal. It's almost death by 1,000 cuts, but just to highlight the major ones, it's you know, overfishing is a huge threat, as we all know, and destructive fishing practices associated with that. Sedimentation due to poor land use practices smothers coral or puts nutrients into the water. Coastal development, you know, destroys nearshore habitat, which are really important to a lot of the keiki fish and lots of other species. So those habitats have been modified heavily over time, particularly around the more developed areas of the state. Invasive species, limu in particular, you can see that picture in South Maui there with all that red limu there. So that has a big impact on not only the health of the reef, but people's ability to enjoy the ocean and the beach. Marine plastics, microplastics have become a huge issue in recent years. And then the umbrella of climate change sits over top of all of that. So, you know, we always have to think about that in the back of our mind, that these systems aren't static and they're being changed pretty dramatically. I'm going to focus a little bit on overfishing because this is an abatable threat that we can deal with and we have dealt with a little bit. But some work we did a few years back where we looked at all of the visual survey data for fishes conducted around the state. Nice paper, Russell's on it, a few other people happened to make the cover of the Star Advertiser. And basically we looked at fish biomass around the state using MOKU, the traditional management boundaries used as kind of a measure of what was going on. And that figure in the bottom right there for our representation there, the big pink bar is northwestern Hawaiian Islands. Those green bars represent MOKU where we've got high fish biomass. And those typically are in places that are remote, like the island of Ni'ihau, north shore of Kauai, north shore of Molokai, the island of Ko'olave, Hamakua Coast. So those places are doing relatively well, and because they're remote for the main part. The bars in orange represent areas with extremely low fish biomass, and they are not surprisingly related with places where there's lots of people. Almost the entire island of Oahu, South Maui, and West Maui also have extremely low fish biomass compared to many other areas around the state. So just kind of shows this gradient of human impact that we can relate to overfishing statewide. One of the best solutions for conserving biodiversity and enhancing fisheries is marine protected areas, such as Molokini, which I'm going to get to in a minute. But the concept is pretty simple. If you don't fish in a place, you're going to get more fish, and those fish are going to be bigger. When you get more fish inside of these protected areas, they build up density dependence where there's too many of them, and they spill over out into the adjacent area where they can be fished. Up and above that, you typically have more and bigger fish inside of these protected areas, and these bigger fish produce a lot more keiki than smaller fish. So a fish twice as big as another fish doesn't produce twice as many offspring. It produces 10 times as many offspring. So these big spawners are really important to the health of the entire ecosystem and the populations. We looked at a number of marine protected areas around the state, and they vary in their levels of protection, from fully protected to areas that have low protection and they're barely protected areas at all. And you can see from this graph, areas with low and intermediate protection don't harbor a lot of fish biomass. Areas that are fully and highly protected do, on the other hand. So Molokini sits up there at pretty much the high end of this graph as far as having high resource fish biomass. It's a well-protected marine protected area, but it's small compared to many of the other areas like Kalapapa or even Ko'olave. So that's why the biomass isn't even higher than that. Okay, so we know marine protected areas are highly effective at conserving biodiversity and enhancing fisheries. And if you look at this map here, it looks like Hawaii is doing a great job of creating protected areas. There's marine life conservation districts, fisheries management areas, all kinds of different areas that are seemingly protected. However, when we look at it, less than 1%, about 1/3% is fully protected of nearshore waters, and only 3% is highly protected. So you can pretty much do anything you want in 88% of the nearshore waters in Hawaii. So even though we know marine protected areas are effective, and it seems like we have a lot of them, they don't cover a lot of real estate, and therefore not as effective as they could be. Okay, now to the question at hand. Molokini Marine Life Conservation District. It was established in 1977. It's got 2 zones, subzone A, which is inside the crater that we often refer to. It's relatively small, about 40 acres, where no fishing is allowed. And then subzone B, which is the area outside of the inside of the crater and sometimes referred to as the backside. That's another 48 acres where only trolling is permitted. So this is a recent photo of Molokini on September 21st at 9:06 a.m. from the DAR camera. And you can see it's a small area. And even though a dozen boats doesn't sound like a lot, that's what you see here. And you see all those people in the water, and it very quickly fills up. It doesn't take a lot. It gets over 300,000 visitors a year. It's one of the most visited marine protected areas in the world, probably the most visited marine protected area in the world on a per area basis because it's so small. It also generates a lot of revenue, over $20 million a year. And that is probably, that is a low estimate. That Molokini is a really unique place, both for Hawaii and globally, and it's probably undervalued as a resource. And as we see elsewhere around the world and elsewhere in Hawaii, we need to value these resources more so they can be better managed. Okay, so as a result of this, several years ago, like 7, 8 years ago now, we initiated a study where we were looking at what effect the human presence in Molokini was having on predators and mobile fish inside of Molokini, led by one of my former students, Alex Filos, myself, and a number of people in my lab and DAR with Adam Wong, Christy Stone, and Russell really helped in this study. And the basic premise of the study, we used acoustic telemetry to look at the movement patterns of fish. So upper left-hand figure there is a palm of someone's hand with a bunch of acoustic transmitters in there. We caught ulua, omilu, sharks, and we surgically implanted these transmitters inside of those fish. Then they swim around, and each tag has a unique code. So each fish is individually identifiable once they swim by these black receivers that you see in the right-hand side there. So the setup is this. In the upper left-hand corner, you can see Molokini. The 3 black dots are the 3 acoustic receivers inside of the crater, and the 4 red dots are on the backside. There was also a series of acoustic receivers around Maui as part of a larger study. Those are the green triangles. And there's also a receiver at Kohalave, that blue square right there. And on the right-hand side, you can see what those receivers look like underwater with a buoy. Okay, so these are some data. This is a gray reef shark, 164 centimeters long, a male. And the way to read this figure is that on the bottom, that horizontal axis there, that's, date. So that's covers about 9 months worth of time. And the vertical axis, the y-axis, is time of day. So the shaded areas represent nighttime. So anywhere from midnight to about 6 a.m. throughout the year. And then again, from about 6 or 7 p.m. till midnight on the top part of that figure. The dots represent detections for each time that fish swam by a receiver. So the red dots show a reef shark was spending most of its time in subzone B, which is the backside of Molokini crater. A little bit of time in subzone A, those black dots there. And also interestingly, those blue squares there show that this fish moved between Kaho'olawe and Molokini. So Molokini is a little bit of a corridor between Maui and Kaho'olawe, and it's really important for a number of species. You see a lot of transient species come by Molokini, manta rays and whale sharks and all kinds of things, and they're using it as a bit of a rest stop as they move around Maui Nui. Okay, so the one species that we really focused on was omilu, or the bluefin trevally jack. And the reason we focused on this species is because it's a daytime predator. You can see from that figure, it looks very different from the gray reef shark figure previously. All those black dots show that the detections for the omilu during the daytime are almost exclusively inside the crater in subzone A. In the evening, it goes out, wanders around, spends some time on the backside in subzone B, but this is really a diurnal or daytime predator. So the question is, how is this and other species affected by all the human presence at Molokini? And for starters, we looked at the residency time of these species. So we've got omilu on the left there, just to the right of that, ulua, white-tip reef sharks, gray reef sharks, and uku, or rosy jobfish. And you can see the omilu and the ulua have the highest residency. They spend the greatest amount of time in Molokini. The gray reef sharks and the uku much less so, but they use it fairly substantially. So do more boats equal more noise? We had a hydroacoustic listening device in the crater, and you can see the bar there, so that's time of day. You can see the peak time for vessels at Molokini is about 8 to 9 to 10 o'clock in the morning. After that, the wind picks up and the boats kind of take off. And that red line there shows noise levels. And you can see there's a strong correlation between the number of boats there and the amount of noise that is made or observed or detected inside of Molokini. So when we look at omilu presence versus the number of vessels, again, on the bottom horizontal axis, there is number of boats, and y-axis or the vertical axis is the presence or proportion of presence of omilu. And you can see omilu are, fairly present and abundant inside the crater when there's a small number of vessels around. When those number of boats increase to about 10 or so, we see a pretty precipitous decline in the presence of omilu inside the crater. And then as you get more boats, they become even less abundant inside the crater. So a strong negative correlation between boats and Omilu present inside the crater. And it's not just Omilu, we found the same for Alua and for Uku. White-tip reef sharks did not show any correlation with the number of boats. But we see the magic number seems to hover around a dozen or so. So anytime you have more than about a dozen boats, 10 to 12 boats inside the crater, it has a significant impact on the presence of these highly mobile predators. So where do they go? This is for Omilu. And if you look at that left-hand bar there, that shows when there's a low number of vessels around, from anywhere from 0 to 8 boats. And you can see most of that bar is blue, meaning those Omilu spend the majority of their time in subzone A. That's their happy place. That's where they want to be, with a small amount of time in subzone B. As the number of vessels increase from about 9 to 16 moderate use, they split their time. They're spending part of the time inside the crater and then part of the time on the backside in subzone B. When you get a large number of vessels there, from 17 to 25, the high vessel intensity, they aren't in subzone A very much, and they aren't in subzone B all that much either. They're pretty much displaced from Molokini when there's that much vessel activity present. So as a result of this work, there were some discussions among DAR and the boat operators about potentially limiting the number of boats, the number of people. Some people felt like there's too many people there. As Russell mentioned before, there was a crowding study where, tourists and people felt like anytime there was over about a dozen boats in Molokini, they felt crowded out. So a little inconsistent with the concept of a marine life conservation district. And then we had this unprecedented opportunity to turn off the tourism spigot that never happened before, and probably and hopefully will never happen again. But it gave us this unique opportunity to, dig deeper into all this work that we've previously done in Molokini and look at it under a whole new lens when tourists and boats were not at Molokini. So I'm going to leave it at that and hand it off to Kevin, and Kevin's going to talk about now the recent work that we've done in the last couple of years as during the pandemic. So take it away, Kevin.
Awesome, thank you, Alan. So this is a photograph of Alan conducting a survey at Molokini, measuring the fish community and biomass. So by doing that, Alan and Whitney were able to gather a big data set that could be compared to all of the information they had gathered in prior years. We were also able to quantify the number of people that were visiting Molokini. This is a figure that was put together by Laura Gajdzik, and she was able to show that for the decade leading up to the pandemic, there's approximately 7,000 to 10,000 people visiting per month. You can see a seasonal cycle with the maximum number of visitors in the summertime. And then right about 2020, of course, the world turned off. We had a very, very strict quarantine policy, and the tourism visitation to Hawaii dropped to almost zero. And then late in 2020, the quarantine was lifted, and then you can see that line start to climb back up pretty rapidly. And as the pandemic started to wind down, the visitation, the tourist visitation to Hawaii actually recovered to pre-pandemic levels. We got some coverage here in the Maui News showing that with the cessation of tourism and several months of very, very quiet period at Molokini, larger fish started moving back into the environment, and particularly into that shallower subzone on the inside of the crater. This is a photograph that Whitney took on the inside And this shows a big school of barred jacks. This is a pretty interesting observation because this species, so this is not the omilu, this is a different species of jack. This species was known from the vicinity, but was not typically sighted on the inside of Molokini. So to have this observation of this large school of jacks hanging around on the inside of Molokini during this quiet period was quite remarkable and indicates that there are species that would like to be using this habitat, but they're not using it because there's so much activity going on a regular basis. So this indicates that when we actually really reduce the human activity because of the pandemic, there was a change in the community structure and a change in the way that organisms were using the habitat that we had not seen in those prior years because there was never a cessation of human activity in all of those prior years. And this is a big school of Littoratus. This is a herbivorous species, and you can see some of the ones down there on the bottom are actually sort of browsing away on the bottom. The herbivores are particularly important in terms of the ecological function that they're playing on the reef, because the herbivores are consuming algae, and the algae and the corals are actually in competition. In order for larval corals to actually settle out from the water column and start growing from their baby phase up into a full-size coral, there needs to be a clean substrate, and that cleaning is done by the herbivorous fishes and other herbivores on the reef. If we take away these herbivores from the reef environment, we're going to push the whole system much more towards algae. That's going to mean much less space for corals to settle out and grow. This is data showing the biomass of fishes at Molokini from years just before the pandemic, during the pandemic, and then the year afterwards. And this is the data that's gathered in the DAR ongoing monitoring program. And so remember what Alan was saying about there being a big variation in the biomass of fishes across the Hawaiian archipelago because of that big change in human use from a very pristine place like the Northwest Hawaiian Islands or Kaho'olawe or Ni'ihau to a much more heavily used place like Oahu. If we look at the data that is shown on this graph, you can see that in the early part in 2018, this is a period of time when we had a normal level of tourism happening, and we had our biomass a little bit below 100 there. When we move along and we see the pandemic period, that's the 2020.4, the biomass has actually gone up to about 150. So that's from less than 100 to 150. That's a large increase in biomass. And then when we go on to the later part of 2020 and then 2021, you can see that it kind of came back down to the pre-pandemic ordinary level. This is happening over just a few years. This is not a time scale when there's enough time for the population to actually reproduce and grow. That's not what's happening here. What's happening here is that there are behavioral changes of these organisms where they are utilizing different places because of the level of human activity. So during those 2018 and 2019 years, we had high levels of human activity. The animals were utilizing Molokini less because of that high activity. And then during the pandemic, when everything shut down, a lot of animals moved into the Molokini area, particularly into the inside of the crater. And a lot of those animals were quite large-bodied, which had this big influence on biomass. This is a graph that is showing which species were driving the changes in the different years. So when you look at those arrows on the graph, they're pointing at a particular species, that's the species that was driving the change in that year. So if you start on the lower left-hand side, in our years before the pandemic, we had very, very high levels of herbivores. That's an image of a nenue, which is an herbivore. So that was kind of the community that was utilizing Molokini during those periods with high tourism. When we moved into the period of the lockdown, when there were no people going out to Molokini, that's the lower right-hand side where you can see that very long arrow pointing at the barjack and the ulua. This is the period of time when there was almost no human activity, and these big predatory animals moved into Molokini and started using that habitat that they had been displaced from before. And remember, these are the species that when we're fishing, we target these species very, very heavily. We call them the resource species because obviously if you catch a nice ulua or a nice big jack, it's a very, very high quality, high table fish. It's a lot of meat that you get. So those are very heavily targeted animals that are more likely to be displaced by human activity than the smaller fishes. And then when we look into the years that are after the lockdown finished, when the tourism started recovering, then you can see us going back into the more herbivorous species and some of the invertebrate eating species that are the that Littoratus unicorn there and the triggers. This is an image of Alan conducting one of the surveys. So Alan is the scuba diver kind of in the lower center of the photograph. And this was during the period after the tourism had resumed. So the lockdown has been lifted, the quarantine is lifted, and we were lucky to get a big influx of tourism to Hawaii to help our economy restart. You can see in this image with this large number of scuba divers and scuba divers, they're creating quite a lot of bubble noise, and there's just a lot of presence of large, what could be interpreted as large predatory organisms in the water. And so this is the type of activity that is most likely to have a displacement effect on the omilus and the other larger fishes of the system. So this is the detection graph from our recent study that we did before, during, and after the COVID pandemic. And you can see here that we're showing a similar pattern to what Alex and Alan showed in their prior study. What we added here is, if you look on the very left side of the graph, right there where the total number of people is very, very low or zero, you can see that there's a huge splattering of points there, and the darker colors there are because there's lots of points sitting on top of each other. So this is what we were able to measure during the COVID pandemic that was not measurable previously, because with the high levels of human activity, that very, very high usage never occurred. So this is where we have organisms, particularly predatory species, moving in to this very quiet habitat during the period of time that we don't have people. So just by way of visual comparison, the photograph on the top left is the one we just looked at from when the tourism had come back really strongly and we had lots of visitation to Hawaii. The photograph on the lower right side is during the COVID lockdown when nobody was able to travel. And that is Alan conducting another survey, and behind him is that big school of barjacks. And that's that species that was known from the area but didn't typically utilize that inside habitat of Molokini. So this is a pretty unusual event for our time, but actually if we didn't have a high level of human activity, it would be ordinary. So this is now looking at a much, much longer time scale. So if you look down on the horizontal axis of this graph, now you can see we're starting all the way back in 2002, and we're coming all the way up to the present. So this is a 20-year time period, 2 decades of data on changing fish biomass. What this graph is most likely representing is changes in the actual population levels of these species. So this is different from the previous graph that we looked at. Now we have a long enough period of time that we can have changes in the population level. We could have reproduction in these species happening, and we can have a lot of fishing pressure happening over that 20-year period. We know that people are always moving to Hawaii. Our population is growing. Our population of human beings is growing. We all love to go fishing. Over that 20-year time period, there has been an increase of harvesting, particularly of the most desired resource fish, which here is represented by the omilu. If you look at the line for the omilu, which is the one that has the black circles, the black dots, over this 20-year period of time, there's a very large decrease in the biomass of the omilu. And this is also represented in other data that Alan and collaborators have gathered on other predatory fish such as uluas. In contrast, if you look at the top line there, that again is our herbivorous fish, that's with the red dots. There has been a small decline over this 20-year period in herbivorous fishes. And there are a lot of herbivorous fishes that are targeted for consumption, particularly the uhus, and quite a few of the surgeon fishes are desirable table fish. So they have been harvested, and their populations have also decreased over this 20-year period. Now, if you look on the very bottom of this graph, we have a mu representing the invertebrate eaters, and then we have a trigger representing planktivore and other functional groups. Those species have not changed very much over this 20-year period, so the harvesting and the targeting of those groups was not intense, and their populations have been relatively stable in comparison to that big drop in population that you're seeing for the predators. So Molokini is really a very, very special place. I mean, everybody who's on this call has probably been there. If you haven't, hopefully you will get an opportunity to go and visit and dive and snorkel there. Molokini is home to lots of really, really important and beautiful organisms. We have the uhu there in the top left, which is such an important fish for fulfilling that ecological function of cleaning the algae off the reef. We have the big roving schools of herbivores like this manini pile that you see here with the yellow fish with the black stripes. Having these very, very high herbivore populations at Molokini is allowing us to have really, really high live healthy coral cover. And Molokini has very, very high cover of healthy coral in comparison to many other places in Hawaii. This is supported by these really, really high herbivore populations. It goes hand in hand. And then, of course, we have some very, very large charismatic organisms, mantas that are occasional visitors to Molokini, and then that's the white-tip reef shark, which is a resident, more of a nocturnal animal that lives regularly in the crater. So in conclusion, we were able to utilize the very unfortunate event of the pandemic to gather information about this reserve during a time that there were almost no people visiting. And that enabled us to gather a data set that you can't gather under normal circumstances. During that period of time of the lockdown, when there was very, very little human visitation, there was a dramatic increase in predatory species utilizing Molokini, in particular that shallow inside portion of the crater. Upon the resumption of tourism, the fish biomass and habitat use returned to the normal levels that had occurred before the pandemic, in line with those higher levels of human activity and human visitation that was displacing those predatory fishes. What this means is that while we know that these non-consumptive uses are not actually taking a fish out of the population the way we do when we catch it for dinner from fishing, the non-consumptive uses can still have effects, such as the displacement of these predators from Molokini. So when we're looking at management of MPAs and MLCDs, and when we're planning the future management of Hawaii's marine resources in 30 by 30, we need to consider not only fishing pressure and pollution, but we also need to consider these non-consumptive effects that human beings can have through displacement. And we've got an incredibly long list of people to thank here. It'll probably take you a little while to read through there, but there were so many people who gave us so much and helped us out so much in order to make this happen. We really, really appreciate all of the support that we got from the community. So thank you, everybody, for being so gracious and generous.
All right, thank you so much, Alan and Kevin, for all of the other researchers that were out there assisting in the study. We already have a few questions coming in, so I'll start reading those out loud, and then whoever would like to answer can jump in. If you do have questions, you can use the Q&A feature at the bottom of your Zoom screen, or again, if you're tuning in from Facebook Live, feel free to leave a comment and it will be relayed to us through the chat. So I'll start with one that came in from Facebook, and it says, really interesting study. How long do the tags last?
Kevin can follow up, but they can last a couple years. So, and, you know, the first go round, we had a fair amount of data, and then we had some difficulties getting back out again. But then when we were able to go back out and recover the receivers, we got some additional data from the fish we had tagged back in, 2016 or 2015. So they last a few years, and Kevin's still got some fish that are ticking away out there now. So they'll continue to collect data for a little while. And it's, it's a great opportunity because those receivers, if they're still out there, we can tag lots of different species and continue to ask additional questions for other species. So yeah, it's a great tool.
Great, thank you. And one in the Q&A, how was Molokini biomass measured pre, during, and post-COVID shutdown?
I guess that's me again. It was two different data sets. We first went out in the early 2000s that Kevin showed. We did an evaluation of all the marine life conservation districts around the state over a couple of year period. So they're done with visual surveys. You basically get in the water, you lay out a transect line, you count all the fish, you size all the fish, you come back, you can, and then you can calculate the biomass based on the numbers and sizes of all the individual fishes. That was the original study. We replicated that during the height of the pandemic and then afterwards. And in addition to that, DAR has a more comprehensive data set that is essentially the same methodology. So visual surveys is the short answer, but there were two complementary data sets.
Great, thank you. And someone asked if it was possible to have the email addresses from the presenters. If you would like to share them, if you wouldn't mind putting them in the chat, and maybe then people can take them from there. Another question, is there any discussion about a reservation system like Oahu's Hanauma Bay?
I could take that, I guess. Hanauma Bay is, accessed from shore. The county manages it. There's other examples like Hanauma Bay that are managed by different entities where everyone is accessing the area by shore. Some state parks are doing it now. Wainapanapa on Maui and on Kauai, the state park in Haiena. as well as also doing reservations. So it's definitely a one way that we've discovered you can start to limit excessive use, right? Haleakala National Park does it on Maui as well. For Molokini, it's not likely we could do that, at least not in the same model, right? Because Molokini is accessed by boat. So these are commercial tour operators that are taking people to Molokini. However, there is a limited number, there's 40 permits. And so there are 40 commercial operators that we can work with, and that's what we're doing. We're working with them. We limited their passenger capacity growth. When there's opportunities to reduce passenger capacity, we do. If they get smaller boats, or some operators are looking at changing their business practices and taking less passengers and things of that sort. So that's what we're encouraging at this point, and we're just looking to see if we can do that voluntarily through, just open discussions, sharing the data, having these discussions with the operators, and having them kind of coordinate their schedules a bit so they're not all there at the same time and things like that. That's how we're working with it right now in Molokini. It's difficult to turn it back substantially because these folks have been in business for quite a while. They've invested a lot of money into their operations. And so it's not like the state can just say, sorry, only half of you can go anymore. But I do think, they are very vested in the health of Molokini as well. And what we found over the last several years is that if we have data like this, we share the data and we talk about it, that it is possible to kind of control the crowding somewhat. So good question there.
Great, thank you. Yeah, there's a few other questions about limiting capacity and limiting visitors and boats in the Q&A. I'll get down to those as well because you touched on some of those points. Someone else asked, how far away from the MLCD can boats fish?
So the subzone A that was shown early on in the presentation is from reefs in, the submerged reef on the western edge of the reserve, across towards the landmass. I think it's called Ninui Point. There's probably another name for it. But anyway, the western edge or the eastern edge of the island. There's a straight line there. You go 100 yards off of that, and then all the way around the backside as well. That's subzone B. You can fish in subzone B, but only by trolling. And to be designated trolling, you have to be making way. You can't just be holding your position against the current. So within subzone B, you can fish. And then once you get past that 100 yards from the inside of the crater area, you can fish like normal. So there's no reserve restrictions after that 100 yards or after subzone B.
Great, thank you. Someone asked, thank you for a very informative presentation. Can you clarify the units in the chart about changes in biomass and trophic level from 2004 to 2022? And then they are also curious about the sharp angles for all species in the comparison from 2020 to 2022 compared to the 2014 to 2018 trend line. Is that just an artifact of the sampling periods?
Those are my data, so I guess I'll get that one. Those are in grams per meter squared. A typical reef around Maui is going to be, something around 60 to 80, maybe 50 to 80 grams per meter squared. So when we're talking about, 100 grams per meter squared or more than that, that's a fairly healthy reef. And you would expect that for a place like Molokini where fishing is not allowed. The questioner is correct that those sharp angles are just an artifact of the fact that there was sampling in 2004 and then not again until 2018 or 19. So it didn't mean to be misleading, but yeah, it's a limited number of data points for that. The data that Kevin showed from DAR is higher resolution in the sense that it was done quarterly as opposed to, that long hiatus between years.
Thank you for the clarification. This will probably go back to Russell. There are a few questions asking about how the number of boats inside Molokini can be better managed, or how can the number of boats be limited? And also touching on, is the goal to decrease the amount of vessels to allow predators to go into Zone A more often? And has it been proposed to limit the amount of visitors to the craters? They're all kind of related. I don't know if you want to touch on all of them, but they're in the Q&A box if you need to go back.
Well, I mean, I'll just reiterate kind of what I said, but first off, the goal is to try to minimize our impacts to the area as much as possible. And part of that is allowing what would normally be there without a lot of people around to be there and to be, you know, practicing their normal behavior, foraging during their normal time in their normal areas. And, you know, it's hard to really anticipate how that percolates down to everything else and why it's important. A lot of people would be like, so why is it that important if you displace the Omilu for a little bit? We're only there for a few hours, they can come back. But, you know, there's a lot of changes that occur and a lot of things that we don't even understand, but the presence of predators does affect herbivores, and it could affect their grazing behavior and things of that sort. So Molokini is a fully protected marine life conservation district. It was recognized as being incredibly unique and special back in the 70s when it was first designated. And so areas like that in particular, I think we should really shoot for trying to maintain as much as possible that natural service, the natural composition of the ecosystem. That said, businesses have been built. A lot of people go there. A lot of people learn a lot of important things on their trip to Molokini. The marine tour operators do an excellent job in educating them about, you know, minimizing their impacts to the environment and, you know, you name it. So there is a benefit to having the tours as well. And, you know, really, like I said, at this point, we don't see this data driving direct and really extensive management at Molokini. We will use it to work with the operators to try to reduce impacts. And we can use our permit and other tools to mandate things if we need to. But instead, we just want to, you know, raise the awareness, work with the operators. We do have a live camera feed on the island that we're working on, and we'll be, you know, releasing through press releases to the public soon. But we could probably list that for you folks as well. Make molokini.live. It's pretty simple website. So we're also using that as a way to just show everyone what's happening at Molokini. You know, people can look at it and go, "Hey, this place is beautiful. You know, really gorgeous. There's a lot of people there at 9 o'clock. Maybe I don't want to go there at 9 o'clock." And maybe through that kind of awareness, people's the customers will start to drive the change as well. So that's kind of what we're hoping. And then just to wrap up the discussion on this quickly, you know, we also want to use this information to guide all future management. So I think Alan or Kevin talked about the 30 by 30 initiative, where we're looking to establish 30% of the nearshore waters in marine managed areas. Well, when we do this, we'll be considering non-consumptive uses along with consumptive uses. So there could be areas where we want to severely restrict, you know, tour operators or other kinds of human activity. Definitely we'll be thinking about it, right? You know, whereas in the 70s when we established these MLCDs, we weren't thinking about tourism at this level. We couldn't anticipate it back then. So you live and learn and you move on, and hopefully we can do a better job as we create new areas in the future as well.
Thank you. Yeah, the Holomua initiative is exciting, and looking forward to see what kind of plans and areas come through with that. There are still a few more questions in the Q&A, but I know it is already 6:30, so I'll take a couple more and then we can start to wrap up here. One of Kevin's slides showed the relationship between number of people in the water and number of fish. It looked like it only took about 50 or 100 people in the water to have the number of fish drop a great deal. What do you feel is a good number of people to have in the water? Carrying capacity, I guess.
The I don't want to put a number on that. I mean, I think that's something that can be discussed between DAR and the operators. There's a pretty strong relationship both with total people as well as the number of boats in terms of that displacement effect on the omilus. So both boat noise and the amount of people is having an effect on displacing those organisms. There's, as Russell was mentioning, there's probably a lot of ways that this can be managed by scheduling and other approaches like that. You could be reducing the number of people in the crater at any given time, but still allowing the same number of people to visit per day, perhaps, so that the businesses are still able, to be sustainable for the boat operators. I mean, nobody's as invested in the health of this place as the people whose businesses rely on it, right? So they really, really love this place and they want the corals to be alive and healthy and have healthy fish populations. So if we can use data like we're getting from this study to optimize things like schedules and people numbers and boat numbers, that can start actually improving the experience of the visitor. If they see more large fishes on their trip, they probably had a better dive, right? Or a better snorkel. So by starting to use this information to optimize things, we could hopefully improve the experience of the visitor. And looking more long term, if it is possible to have sort of a higher end experience, it's possible that the operators could have a higher price for those experiences, and then you could make the same amount of money with less visitors. So, just lots of ideas could come out of this to try to optimize that management.
Great, thank you. There are a few more Q&As open. I'm going to let y'all type those answers while I'm closing out here. So whoever would like to take an effort at those, please feel free. Otherwise, I'll go ahead and close us out with some fun announcements from Maui Nui Marine Resource Council here at the end. But I just wanted to give a really special thank you and appreciation for all of you researchers and scientists for taking the time to be here and in the different time zones and everything. So thank you so much for sharing your data and sharing it in a way that is fun and easy to understand, especially about such an important place here in Hawaii. So thank you all again, and we really look forward to seeing more research from you all in the future as well. Again, our next Know Your Ocean Speaker Series event will take place on Wednesday, November 2nd at 5:30 p.m. via Zoom, and we'll be hosting Dr. Kim Felinsky. She will be presenting her research on Olowalu and include updates on nearby Hui Okawaiʻola water quality monitoring sites. To sign up for this talk and to learn more about future presentations, please sign up for a free subscription to our monthly e-newsletter, Reef and Brief, at mauireefs.org. Supported by the Maui Visitors Bureau and funded by the Hawaii Tourism Authority through the Destination Management Action Plan, we invite the hospitality industry to take part in a new pilot program from the Maui Reef Friendly Landscaping Coalition to implement chemical-free landscaping on a portion of their property. The program uses a biological soil amendment produced by Environmental Solutions Maui, located upcountry, to restore soil microbiology to support plant health. This effort is supported by Maui Nui Marine Resource Council, Environmental Solutions Maui, Beyond Pesticides, West Maui Ridge to Reefs Initiative, and Edaphic Consulting. If you or anyone you know is looking for reef-friendly landscaping resources, please visit mauireefs.org or email info@mauireefs.org. Some of you may be familiar with the Healing Land, Healing People film, With support from the Maui CARES Act program, Maui Nui Marine Resource Council hired Leah Warshawski of Inflatable Film to document the program that MNMRC sponsored, which used CARES Act funding to employ 70 unemployed or underemployed Maui and Molokai workers on 7 environmental and cultural restoration projects with small local nonprofits. The full-length film, Healing Land, Healing People, is now available to view on our YouTube. Just search Maui Reefs and you can view it. It got a lot of awards at film festivals this summer, and now it's available for you to view online on our YouTube. Very moving and exciting film to see. Highly encourage everyone watches it. You can support continued water quality monitoring by adopting your favorite beach or by becoming a trained water quality sampling volunteer. This community-based water quality monitoring program, launched and conducted by Maui Nui Marine Resource Council, the Nature Conservancy, and West Maui Ridge to Reefs Initiative in partnership with the State of Hawaii Department of Health Clean Water Branch, tests ocean water quality every 3 weeks at 29 locations in South and West Maui. Visit hui'okawai'ola.com for more information. When you donate to Maui Nui Marine Resource Council, you'll also be supporting our work to improve the ocean water quality in Maalaea Bay. Our projects include our oyster bioremediation project, which has caged oysters in the bay, filtering sediment and other pollutants out of the water. We've also planted non-invasive, deep-rooted, and drought-resistant vetiver grass to stabilize the slopes of Poakea to reduce sediment runoff in Maalaea Bay. If you've missed some or all of tonight's presentation, you'll be able to view it on Facebook Live as well as all of our past presentations, as well as on our Maui Nui Marine Resource Council Facebook page. It will also be posted on our YouTube channel, Maui Reefs. And last but not least, we want to thank our sponsors and supporters who are making our work possible. And we also wish to thank individual donors like you for showing up and supporting webinars like this. Mahalo for making a difference. Thank you all for joining us again. Have a wonderful evening, and we'll see you in November. Aloha.